started. Hello, everyone. Welcome. Thank you so much for joining this month's Fireside Chat in honor of Bone Cancer and Sarcoma Awareness Month. The Fireside Chats are brought to you by the Office of Community Outreach and Engagement at the Masonic Cancer Center at the University of Minnesota. All right, and here's the Office of Community Outreach and Engagement. And so the Office of Community Outreach and Engagement serves as a bridge between the Cancer Center and communities across the state of Minnesota. Our team's mission is to reduce the burden of cancer in Minnesota by engaging communities and providing them access to knowledge and information on the latest cancer research, cancer prevention, the importance of screening and early detection, treatment options, survivorship, as well as clinical research opportunities. My name is Amna Hussein. I am the Community Engagement Coordinator on our team, and on behalf of our team, thank you all so much for joining us today to help us raise awareness about bone sarcoma. Bone sarcomas are named the forgotten cancers because of how rare these cancers are. Sarcomas account for only 1% of cancer diagnosis across all age groups. This month, we want this forgotten cancer to become common knowledge for all of us. Awareness and information are key to early detection and moving research forward in this field. So today, we are joined by Dr. Logan Spector. Dr. Logan Spector is a University of Minnesota researcher and professor. His research focuses on the causes of childhood cancer with a focus on childhood leukemia, bone sarcomas, and hippotomeblastoma. His work includes both traditional and genetic epidemiologic approaches. He works in collaboration with colleagues and trainees locally, nationally, and internationally through the University of Minnesota, Children's Oncology Group, and Childhood Cancer and Leukemia International Consortium. He is currently the chair of the Childhood Cancer Leukemia International Consortium, which pools epidemiologic studies of pediatric cancers across the world to better ascertain their causes. Today, Dr. Spector will go over the epidemiology of bone cancer in children and adults, including incidence and outcome. He will then go on to briefly discuss what is known about the causes and describe the research ecosystem for these rare cancers. So we will be recording today's session and we will post it on our YouTube page later this week. Before we begin, I would like our audience to get familiar with using the Q&A feature, which is located on the bottom of your screen. During today's presentation, please use the Q&A feature to submit any questions that you have. All right, and without further ado, it is now my pleasure to introduce to you all Dr. Logan Spector.
Thank you for the kind introduction. And I'm really pleased to be discussing bone cancer, which I've been studying for nearly since I arrived at the University of Minnesota. Actually, my 20-year anniversary at the U is coming up on August 5th. So it's blowing my mind that I've been here this long. And I'll just gonna take a little aside to let you know how I got interested in bone cancer, more properly bone sarcomas. When I arrived here as a postdoctoral fellow, I was assigned to write a chapter on pediatric cancer epidemiology. I arrived here having done a dissertation on pediatric leukemia and knew nothing at all about the solid tumors. But in writing that chapter, I got a crash course in what caused most other solid tumors in kids. And the two cancers that struck my interest the most for different reasons were osteosarcoma and Ewing sarcoma. And I study them to this day. So I will now chat about bone cancer with you. So just to give you an overview, we will just go over a little bit about the molecular biology. I promise you it won't be too technical. I know that this audience is mostly community members rather than scientists. And then secondly, I should add that if you have questions that come up as I'm talking, feel free to put them in the Q&A or chat, and I will try to answer them as we go rather than waiting till the end. Then, because I'm an epidemiologist, I will talk to you about the descriptive epidemiology of bone cancer in children and adults, including how often it occurs, how well people survive, and then a little bit about what happens in the long term to survivors. Also, because I'm an epidemiologist and this is my main research interest in bone sarcomas, what causes them. And then lastly, I'll talk a little bit about the research ecosystem for bone sarcomas in particular, and sarcomas in general. So first, what is a sarcoma? All right, it's a scary word that not a lot of people know. I mean, it even sounds scary, and it is. Sarcomas as a class of cancer are all rare, and they have in common their tissue of origin. So they appear in different tissues, but what these tissues all have in common is where they come from in the developing human. All right, so immediately you'll notice that there are some of these sarcomas in red and some in blue. So the ones in red are so-called soft tissue sarcoma. Soft tissue is basically any tissue that is not bone. Bones, as we all know, should have some structure. Without them, we would fall apart. So osteosarcoma, it has the prefix osteo, which clearly means bone. Ewing sarcoma is not too clear to people what that means. It was named after the pathologist who first described it, James Ewing. And Ewing sarcoma can actually appear in or on the bone or in the soft tissue, but the vast majority of cases are in or near the bone. So we typically group it with bone tumors. There is a third type of bone cancer. Actually, it's chondrosarcoma. So it's actually a cancer of an adjacent tissue, cartilage, which, as you will probably know, is not really hard. It's more soft with some a little bit of rigidity, but nevertheless, because it's usually in between two bones to keep them from rubbing against each other, chondrosarcoma is classed as a bone tumor. I will not be talking about chondrosarcoma today for a couple reasons. One reason is it's much less common than osteo or Ewing sarcoma. And then the less admirable reason is I don't know too much about it and can't speak about it with any authority. Excuse me. And then I mostly won't be talking about any of the soft tissue sarcomas, also for these reasons. So the bone sarcomas are often studied together or in contrast, especially osteosarcoma and Ewing sarcoma, because they have some commonalities that are interesting and some differences that are informative. All right, so first thing to note is osteosarcoma, 70% occurs in the long bones of the lower limbs. All right, and if we were to zoom in on where in these bones it occurs, it is mostly around the growth plates. And so bones grow by not by expanding from the middle, but by building onto the ends. And osteosarcoma mainly occurs where that growth is occurring, in the young. Whereas Ewing sarcoma, the tumor tends to appear kind of proportionally across the skeleton. And so, you have about 22-25% of your bone mass is here, and that's where your Ewing sarcoma occurs, and similarly with chondrosarcoma. So first interesting difference is this predilection of osteosarcoma for the long bones of the lower limbs, where you get most of your height during puberty, Also, most of the risk for osteosarcoma in children is around the time of puberty. And so first clue to cause is that it coincides very much with puberty and the pubertal growth spurt. All right, I mentioned the commonality in sarcomas is the origin, all right? Really, there's 3 layers in an embryoblast, which is 3 weeks after conception: ectoderm, mesoderm, and endoderm. And as you can see, different organs and also different pieces of organs or linings of organs come from these different layers. The mesoderm gives rise to skin, the lining of blood vessels, muscle tissue, connective tissue, and these other organs. And if you go a little bit further into developmental biology, the mesoderm has a special layer called the mesenchyme. All right, and the mesenchyme or mesenchymal layer is what develops into muscle and connective tissue. All right, and so sarcomas are considered mesenchymal tumors, meaning that they all derive from the mesenchyme or are in organs or pieces of organs that derive from the mesenchyme. Okay, so I searched for a picture of evil twins and it came up with this picture from The Shining. And in popular talk, you know, there's an evil twin and a set of twins. But I like to think of osteosarcoma and Ewing sarcoma as twins where both are evil, even though they have these contrasts. So first contrast is osteosarcoma has one of the most disordered genomes in the cancer of any cancer. And Ewing sarcoma is comparatively very simple. This is a graph of the number of mutations per length of DNA in different pediatric and adult cancer cells. As a rule, adult cancers have more mutations. Likely that's because they've had decades to acquire them, whereas in pediatric cancers, it isn't that many years to have acquired them. But you will notice that osteosarcoma is on the right-hand side of the curve. And here we're just talking about single nucleotide polymorphisms, meaning a 1 DNA letter change, which aren't actually the major form of cancer mutation in osteosarcoma. Ewing sarcoma is in the middle of the pack here, but it's this graph that actually displays just how complex osteosarcoma is in terms of big chunks of DNA getting mutated. We call those insertions, deletions, translocations, or inversions among the pediatric cancers. And if we graph this in adults, it would also be true. Osteosarcoma has the most complex genome. I like to call it a Frankenstein genome because there are pieces of chromosomes that have been taken apart and put together in odd ways, pieces missing, pieces duplicated, to the point where it is a major issue trying to figure out which of those somatic mutations is actually causing the osteosarcoma to behave the way it does. By contrast, you do not even see Ewing sarcoma on this graph, and that's because it is one of the simplest tumors around. There is a single genomic translocation called EWS-FLY1. You take a piece from the EWS gene, you take a piece from the FLY1 gene, they get fused together inappropriately. It creates a transcription factor, which hunts out pieces of the genome and causes some genes to go up in expression and some genes to go down. And that single change to the somatic genome is enough to cause Ewing sarcoma. A little bit further, I will talk about survival. And this is, again, why Ewing sarcoma and osteosarcoma are often contrasted, and puzzlingly so. Osteosarcoma has a very complex genome where we don't know really what's driving the phenotype. Ewing sarcoma has a simple genome where we have figured it out, but that simple change causes a terrible transcriptional program. And both of these tumors are similarly difficult to treat. They both have similar survival rates, despite these differences in the molecular biology. So moving on to incidence, you'll note that pediatric cancer is extremely rare, and when you graph it versus the adult form, it barely registers. In fact, cancer in people under 20, which is how NCI defines pediatric, is only 1% of the total burden. Within the pediatric age range, you can see there are also differences in risk by age and different classes of tumors that occur by age. The teal or sea green or whatever you want to call that is combined bone and soft tissue sarcoma. So while these are more common as a share of all cancer in children, they are still very rare. And remember, this is bone and soft tissue together. So 10 years ago, I was surprised to learn in putting this talk together. I published with colleagues a review, which remains my most highly cited publication, The Epidemiology of Sarcoma. It's open access, so you feel free to download it. It certainly is in need of an update, especially with the genomic findings that have happened in the last 10 years. But I grabbed some slides from here because the distribution of tumors hasn't really changed. So among all sarcomas, bone tumors are about 13%, with the rest being soft tissue. And then within the bone tumors, I already mentioned the major types are osteo and Ewing sarcoma. I guess chondrosarcoma is fairly common in older adults, but again, since I don't study it, you're not going to get a very informed talk from me. And as the pie graph implies, bone tumor incidence is quite a bit lower than soft tissue sarcoma. And when it's plotted this way, you can't really see what's happening. So now we'll zoom in on these two bone tumors that I'll be talking about. So osteosarcoma incidence is what we call bimodal. There is an early life peak that is, got the highest incidence, and then a low but steady incidence, and then a much smaller later life peak. This graph also distinguishes between primary bone cancer, meaning that it's the first instance of a bone cancer, and subsequent, meaning that it's a second cancer. All right. And you see that the black line here grows with age. All right. One of the known risk factors for osteosarcoma is prior chemotherapy and radiation. And so what you're seeing here is osteosarcoma as a second cancer among people who were previously treated for cancer. Ewing sarcoma is also displays this early life peak, peak of an incidence around puberty, but then the incidence disappears to near nothingness past age 40 or so. So there is no late life peak as in osteosarcoma. Last year or the year before, colleagues and I published on the international incidence of osteosarcoma and Ewing sarcoma. And here's another contrast that is interesting. We found roughly the same rate of osteosarcoma and across the world. and in the very same pattern. So no matter where we looked, and this is in International Agency for Research on Cancer data, there was a peak around the time of puberty, and then a late life peak as well, although it was more or less pronounced depending on the area. In contrast, Ewing sarcoma has some very distinct patterns of incidence across the world. So here we're using the same data set, but looking at Ewing sarcoma, and we classed the populations according to predominant ancestry. It has long been known that Ewing sarcoma affects kids. In the U.S., it's known that it affects kids with European ancestry far more often than kids with other ancestries. In particular, it is nearly absent in kids with significant African ancestry, but also intermediate incidence in Latino and East Asian. Here we took a broader view with a lot more ancestries. One interesting fact that came out is that Pacific Islanders or Maori, who are related, had the highest rates in the world, followed by Middle Easterners, and then Europeans. Another thing to note, there were something like 200 million kids looked at in the Chinese data, and so few cases that you can see they're here at the bottom. So a real clue that ancestry is one of the driving factors behind Ewing sarcoma incidence. All right, I'm moving on to survival. And we know a lot about survival of these tumors from the Surveillance, Epidemiology, and End Results Program, or SEER. And in this paper, some colleagues at the National Cancer Institute looked at the change in survival for osteosarcoma over the years. So starting in the 5 years, 75 to 79, and ending in the most recent era of data available when they published this, 2010 to 16. And then they divided it by ages, because there is an association of age with the amount of survival. And as you can see in 75, 79, the 5-year survival was awful at 20%, and has steadily improved. In the most recent era, overall, the survival is roughly 80%. I actually need to go back to the paper. I'm not sure if this is overall or in the group of people with without metastasis at diagnosis. So I apologize, and I'll clarify that. We see a similar picture in 10 to 24-year-olds. Actually, in 25 to 29-year-olds, there hasn't been a huge degree of, 25 to 59-year-olds, there hasn't been a huge degree of improvement, albeit the survival was at a better point, and then quite a bit of improvement over age 60, although survival is topped out at 40%. All right, I mentioned metastasis at diagnosis. So I think many of you, if you listen to these fireside chats, know that metastasis means that the cancer has spread from its place of origin to another area of the body. All right. This is also a good time to mention that when we say bone cancer, we mean that it originated in the bone. Bone happens to be a fairly common organ for metastasis of other cancers. So if a cancer cell starts in the liver, and the cells break off and they land on the bone and grow there, that is not bone cancer. That is a liver cancer which has metastasized to the bone. And so I just want to be clear, here we are talking about metastases where a piece of the osteosarcoma has broken off and landed elsewhere in the body and established a colony, if you will. Metastasis is almost always bad news. And so you can see that in the early years, it was essentially 100% fatal. And with chemotherapy and radiation and surgery, it has risen to 40% survival after 5 years, which still makes it one of the least survivable pediatric or adult cancers. And especially in older people, if the cancer is metastasized at diagnosis, it's a worse prognosis. I should add that metastasis at diagnosis seems to be driven mainly by biology, and I don't have time to go into why that is. It happens in about 15 to 20% of osteosarcomas. Over 60 metastatic osteosarcoma has a very poor prognosis. All right, and then this is a similar look at survival for Ewing sarcoma, albeit their graphs were put together more conveniently for me. So in panel A, you see survival for non-metastatic Ewing sarcoma, meaning that that the patients didn't present with metastasis. That's about 75% survival. For metastatic, it's about 30%. And actually, it's a higher proportion of Ewing sarcoma patients present with metastasis. To my recollection, it's 25 or 30%. So it is a real problem. Survival is better in people under 20 at diagnosis versus over 20. Survival is better if the cancer appears in an arm and a leg versus the trunk of the body. And also, perhaps not surprisingly, the larger the tumor at diagnosis, the worse the prognosis. And all of these figures are what we call multivariable, meaning that they're controlling for the others. So in terms of ranking the risk factors for a poor outcome in Ewing sarcoma, metastasis at diagnosis is the greatest factor. Size of tumor would be next, and then location and age are least. All right, so as you saw, there was improvement in survival over the years in osteosarcoma, and a similar graph would obtain for Ewing sarcoma. And while that is brings hope that more kids and adults affected by osteos and Ewing sarcoma are surviving, that survival comes at a cost. So there are studies, including the Childhood Cancer Survivor Study, which originated at the University of Minnesota, by the way, which are tracking the health of survivors of pediatric cancer. To date, there have been very few studies of survivorship in adult. Let me back up. The Childhood Cancer Survivor Study is studying people who are presently adults who survived a cancer in childhood at least 5 years. All right. There are not, to my knowledge, survivorship studies of people who developed bone sarcoma as adults. And so I just want to be clear about that distinction. So one thing you notice, if you've been reading this graph while I've been talking, this is the survival curve for the general US population. And in other words, if you start with a group of people around the time that kids are diagnosed with osteosarcoma and follow them, 98% will be alive in 30 years. Whereas mortality, even up to 30 years after an osteosarcoma diagnosis, is much higher. in the survivor population. And the causes of death are elevated for many different reasons. So this is what we call a standardized mortality ratio. That is how much greater or how much lesser is the death rate due to either all causes of death or specific causes of death in one population versus the general population. And so survivors of osteosarcoma have roughly 6-fold rate of death versus the general population. The rate of death due to a second malignant neoplasm is about 8-fold, due to cardiac or pulmonary reasons 3 to 4-fold, and then non-significant reasons, a non-significant increase for external causes, meaning say accidents. The CCSS also compares, mortality is important, of course, but there are many things that can affect somebody's health without leading to death. And so they compare these things in osteosarcoma survivors versus their unaffected siblings or versus other survivors. I'll just focus on the sibling data. Here we have a relative risk, meaning how much more common was a health condition in the survivors versus the siblings. And you can see that they had a 3-fold greater risk of more than 2 grade 1 to 4 chronic health conditions. And for some things like functional status or activity limitations, they were 7 or 8-fold more likely. I saw a hand raised.
Yeah, it was put down, so you can.
Okay, very good. In terms of life attainment, there can be kind of psychosocial effects. So a higher probability of never having been married, a lower educational attainment, a higher probability of unemployment, and being on public insurance. So the so-called late effects are not just health-wise, but also, I guess, life-wise. This is from the same study, but looking at adult survivors of Ewing sarcoma. And I won't belabor the point, but if you look at the relative risk, and this is compared to siblings, you see double the risk of any grade 1 to 5 chronic health condition, a 22-fold risk of congestive heart failure versus their siblings, which is due mainly to chemotherapy that is used to treat them, a 126-fold risk of leg lengthening, which obviously has to do with operating on the leg during childhood. All right, so moving on to causes, which is really what I study. Again, in this free open access review that I wrote, which again is 10 years out of date, but not a lot of changes in the non-genetic epidemiology. We kind of laid out the different causes based on strong evidence, suggestive evidence, no evidence, or too little data. This could use some updating, but you're welcome to take a look at it. I already mentioned the biggest clue to the cause of osteosarcoma is its pattern of occurrence. Just to delve a little bit more into that, this has been known at least since the 50s, because this graph is from 1958. On the larger axis is the incidence of osteosarcoma in males and females. And down here is a graph of the pubertal growth spurt. So females have an earlier growth spurt, but it is less intense. Hence, on average, females end up shorter than males. Males have it later, but more intense and end up taller. And these are so clearly reflective of each other that it's been very obvious for many decades that there is some sort of relationship between osteosarcoma incidence and bone growth. And so among the known risk factors for osteosarcoma, I already mentioned that irradiation and chemotherapy are established causes and responsible for that growing share of patients over the decades that are due to that are second malignancies. So the first cancer didn't have to be osteosarcoma, but these things will cause it as a second cancer. Secondly, although it is fairly difficult to study because humans don't vary as a percentage of the average height. We don't vary in height as much as you might think compared to, say, dogs. You think of a Chihuahua and a Mastiff. The Mastiff is 5 or 6 times, maybe more, the height of a Chihuahua, whereas you very rarely get humans that are twice the height of another, and only really on the very extremes. Nevertheless, there is some evidence that the taller somebody is, or the taller they are genomically destined to be, the higher the risk of osteosarcoma, but it's not dramatic. On the other hand, with the advent of sequencing technology, we now know, and I am on this paper buried in the authors, but we now know that up to 30% of people with osteosarcoma have a pathogenic or likely pathogenic variant in a classic cancer susceptibility gene, meaning that they have a defective or aberrant copy of a gene that, when mutated, can tend to cause cancer. So just to put that in lay terms, about 1/3 of patients have a mutation in a bad news gene, is how I would put it. And I'm just going to mention some of the genes, but the most famous one is TP53, which is also associated with Li-Fraumeni syndrome. In another study of mine, I don't really expect you to absorb this graph, but maybe the title, we compared the genomes of kids with osteosarcoma to their parents and found that nearly half of the TP53 mutations in the kids were not present in the parents, meaning that it was a de novo mutation. It had occurred in the kid and they didn't inherit in the parents, from the parents. It was a new appearance. So a still developing story in osteosarcoma is that a share of them are due to new mutations. All right, moving now to the causes of Ewing sarcoma. I already mentioned that there are some very stark and robust differences in incidence based on ancestry, with Pacific Islanders, Middle Easterners, and Europeans having the highest incidence, Latinos having incidence in the middle, and East Asian having some lower incidence, and then African or African American, the disease being nearly completely absent. And so ancestry is in fact the strongest risk factor for Ewing sarcoma, and we at the University of Minnesota are pursuing some studies to try and understand why that is. This is also true in the United States. This is data, a graph I generated for a grant application in the SEER data. Incidence is highest in non-Hispanic whites, intermediate in Hispanic or Latino, smaller still in Asian, and then very low in non-Hispanic Black. There have been genetic studies of Ewing sarcoma. Here we're depicting not pathogenic variation, but single nucleotide polymorphisms, meaning very common genetic variants that any one of us might share, and which mostly are just part of the normal human variation. But at these 7 or 8 locations, these SNPs, as we call them, do seem to confer a higher risk of Ewing sarcoma. Interestingly, if you map the frequency of these variants in different populations, there's not a clean story where the high risk populations all have the higher risk variants of these genes. So we don't think that these genes that are discovered to date are explaining these ancestry related patterns of occurrence. More recently, there has been sequencing studies which are looking again at mutations in the bad news genes, similarly to how it was done in osteosarcoma. And surprisingly, or at least it's surprising to me, because there's never really been a familial syndrome that's associated with Ewing sarcoma. 10% of cases have a pathogenic or likely pathogenic variant in a DNA damage repair gene. So that's what this is depicting. And here you can see it's not a large share of the cases. This would be about 5% of cases versus about 1.5% of controls that have a mutation in one of these 5 genes. Lastly, there hasn't been a lot of what I'd call environmental or non-genetic epidemiology of Ewing sarcoma, but oddly, based on the very small literature that exists to date, there is an association of prior hernia with Ewing sarcoma. Now, I never set out to become a hernia researcher, but it is kind of interesting. Hernias are considered developmental defects. In other words, there was a failure during development to kind of separate organs. That's what a hernia is. You got one piece of the body poking inappropriately into another, or in the case of an umbilical hernia, poking kind of out the belly button. But there does appear to be a consistent higher risk for Ewing sarcoma based on having had a hernia. And mainly it's just an interesting side note, but it may indicate that Ewing sarcoma has a in utero origin. So very briefly, I'm talking about the research ecosystem. As you might imagine, for rare cancer, there is not a huge research environment like there is for common cancers. On the other hand, the rarity compels collaboration. So there is a clinical trials organization called SARC, which conducts clinical trials for various forms of sarcoma, both adult, mainly adult. In the childhood space, the 800-pound gorilla is the Children's Oncology Group, which is a federally funded cooperative clinical trials group. These groups do work together because age 20, which is the dividing line for pediatrics, is arbitrary, and there are some good scientific as well as kind of numerical reasons to be conducting trials across arbitrary age boundaries. There is also CTOS, which is the Connective Tissue Oncology Society, which is the more basic science, biologically oriented organization which studies sarcomas. I always wondered why they didn't call themselves the Sarcoma Society, but SS would probably be a tainted abbreviation. So they went with CTOS. We have several raised hands now.
Okay. Do you want to type in your questions in the Q&A? for those of you who have their hands raised. Thank you so much.
Thanks. And I left off this slide that there's also some tumor-specific organizations. The one that I should have put on here is the fairly newly formed Osteosarcoma Institute, which, as it sounds like, funds research for osteosarcomas. Actually, I don't know that there's a major organization specifically concerned with Ewing sarcoma. As far as funders, we have two major funders and then lots of foundations. So the NIH is the major funder in the US for really all biomedical research. But as of late, the Department of Defense also has a strong program and has particular interest in sarcomas with several grant mechanisms that are meant specifically for sarcomas. You might ask, why is the Department of Defense involved in funding cancer research? And the short answer is, it is a lot easier for Congress to insert funding into a defense bill. And so the Department of Defense over the years, even though it makes very little sense, I guess, politically, funds cancer research. There are some sarcoma-specific national foundations, like Sarcoma Foundation of America. There also are local ones. So Rain and Sarcoma, who some of you might have been at their party in the park last night at Como Park, is based here in Minnesota, and gives mainly to Minnesota institutions and more recently to the Midwest. But that's about it for us. I'm not naming every possible foundation, but it is a fairly thin number of organizations that are concerned exclusively with sarcomas, much less bone sarcomas. So that's it for my talk, and I am really happy to answer questions now.
All right, well, thank you so much, Dr. Spector, for just your very interesting and informative presentation today. So before we jump into our discussion, I'm just going to share our campfire.
So I see one question from Aaron McGuire. Is the origin of Ewing sarcoma in the scapula considered extremity or axial? I think scapula is this, right? So then that would be axial, because it's in the on the main trunk of the body.
All right, and then also, so knowing that bone cancers are rare in adults, how can we participate in research to improve early diagnosis and treatment? So this question came through the registration.
Well, if one is a patient, then likely you would be offered a clinical trial, whether that's a phase 1 trial, which is a small kind of safety focused trial, or a larger one like phase 2 or 3, which are meant to actually evaluate the efficacy. Chances are that you would be offered that. If a pediatric patient is diagnosed, the Children's Oncology Group has a protocol called the Project Every Child. And so anyone who's diagnosed with cancer under 20 would be eligible to join. And there you give a little bit of information, a DNA sample and a tumor sample. And the scientists take that and can extract a lot of information from it. For those who are not patients, there isn't a large amount that one can do as far as participating in research, but if you are ever contacted to be a healthy control in a study, we would appreciate it very much if you would participate. we have a hard time getting people who are unaffected by disease to participate in research, and we really need them to know what the difference is between those who are affected and those who are not.
Okay. Another question just came through. What do you attribute the higher 5-year survival rates in osteosarcoma in the most recent decade, when the standard of care is still MAP, which has been used in many decades?
That's a really well-informed and cogent question. I think that the survival rate in the most recent era compared to even a few decades ago is not that different, but there are improvements in limb-sparing surgery that could explain that. And then, you know, as far as the questioner mentions, the basic backbone of therapy for osteosarcoma has not changed in decades. And so I certainly agree it doesn't on the surface make sense that survival would improve when the treatment hasn't really changed. So the best that I can think of is that the other practices around treatment have improved.
Okay. And then, so I know you talked about the genetic component. Is genetic testing recommended for only individuals with a family history of sarcoma?
It should be, but it isn't necessarily the practice. I mentioned that, you know, in my findings, there's a small but noticeable proportion of osteosarcoma patients who have a de novo mutation. They would not have a family history because they're the only ones in their family who carry this mutation. And so I think and then more generally, we found a third of osteosarcoma patients have a mutation in a bad news gene. Because of this, I think the field is moving towards recommending universal germline sequencing for sarcoma patients in order to detect that share. But it's still a debate about whether to offer that or who's going to pay for it. But certainly things are trending that way.
Okay, and then so what aspect of research gets you most excited about treating these rare cancers?
Well, I am excited by the innovations in targeted therapy and immunotherapy. You know, even though that's not so much my area, targeted therapy means instead of using chemo, which can affect both the non-cancerous and cancerous cells, you know, which is why you get a lot of sickness or hair falling out or mouth sores, etc., and chemotherapy. Targeted therapy just tries to treat the cancer cells by focusing on the things that make the cancer cells unique. Immunotherapy, as the name implies, tries to turn on the immune system to attack the cancer. These are exciting developments, not what I do. The things that I am most interested in is just the incredible amount of information that is unleashed by modern sequencing technologies. And that data just gets richer and richer. So we're going to get a better biological portrait of how these cancers work. And I hope that that's going to give us a better idea of how to attack the cancers.
If there are no other questions through the Q&A, I think we'll only have time for just our last question for Dr. Spector. So Dr. Spector, tell us what are the key takeaways from your presentation today for the audience?
I think the key is that bone cancers are rare and, you know, thankfully, but that that has spurred the scientific community to work together We really have to. There can only be 1 or 2 clinical trials of these cancers at a time. That the therapy and has been roughly the same for the past 40 years, and survival has therefore plateaued. We do urgently need research into how to improve the outcomes for these cancers. I also wish to enable early detection, but it's a difficult prospect because of the rarity of the cancer. But I'll just say this: osteosarcoma, even though it's rare, is as complex as any other cancer, more so. And so the resources needed to crack its code are as intense as a more common cancer. And Ewing sarcoma, although it's comparatively simple in its genetics, the downstream effects of the mutation that causes it are themselves very complex. And similarly, we need to throw a lot of resources at studying these cancers. So I don't know if this is an optimistic note or not, but we need more resources to study these rare cancers.
Well, thank you so much for that. And unfortunately, we are out of time, so we'll have to leave it here. Dr. Spector, thank you so much for taking the time to join us today to help us raise awareness about the forgotten cancers. So you will all receive an email shortly following today's session with a link to complete a quick survey. We would really appreciate it if you could just take a few moments to provide us your thoughts about the session, because they will help us shape future programs. After completing the survey, you will be entered to win a swag bag filled with goodies from the Masonic Cancer Center. The $5 Amazon e-gift cards will be emailed out in a few days. And so please follow the Masonic Cancer Center on all of our social media platforms to stay up to date on our future announcements and events from the Office of Community Outreach and Engagement. This will be our last Fireside Chat this summer, so make sure that you stay connected with us on social media to find out when the next Fireside Chat is happening. Well, thank you everyone so much for joining us. I hope you all have a wonderful rest of your day. Take care.
Thank you.
Thank you so much, Dr. Spector.